Amino acids cannot join by themselves together. They require ribosomes to create the tether. All the protein chains get made according to instruction, carried by mRNA in peptide bond construction.
I know how much you're going to miss these metabolic melodies when they're gone, but we're going to start our last section with proteins and nucleic acids today. Our proteins are composed of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. And when you're looking at them, they have that amine group and carboxyl group with the radical in the middle. This is an amino acid, which is the monomer, and their shape and the complexity of the protein determines what their function is. Most are insoluble in water, which is also great because if you think about it, you'd walk out and your hair would disappear otherwise. Let's go over to our functions. Our functions, we said this is the workhorse. Everything is dealing with a protein. They are biological catalysts or enzymes. They move material through cells through the use of microfilaments and microtubules, and actin and myosin in our muscles actually are made of protein, so every movement we have is that. You store material and move things through hemoglobin. It is part of your hair, your nails, all of your cells. It's the hormones and also the antibodies when you're looking at fighting off some type of disease. So again, remember, protein is essentially our workhorse of the organism. It's involved in everything. Let's look at what the protein is made of. We said the monomer is an amino acid, and if you look at that upper right-hand corner, it shows the amine group, the radical, which changes those 20 different elements or components, and then the carboxyl group. When you are going through dehydration synthesis, you take off the OH or hydroxyl group from the carboxyl and the H from the amine group, and you end up with getting that dipeptide or polypeptide. There are several polypeptides that I want you to know. First one is insulin, the hormone that breaks down glucose. We're actually going to make it later on this year. It only has 51 amino acids. It is also the smallest naturally occurring protein. The other three that I'd like you to know are hemoglobin, HGH, and interferon. Hemoglobin is what carries our oxygen, and it's important when we talk about later on sickle cell anemia. The hemoglobin doesn't carry enough. Also HGH, human growth hormone. If there is some genetic alteration, you're not creating HGH on your own, we can now synthetically create it. And lastly, interferon. This was a drug that we thought would be able to pretty much take care of any disease, the little drug that could. Unfortunately, it didn't live up to our expectations. Now, I've mentioned there are different levels of complexity. Protein could be primary, which is just a straight chain of amino acids. Secondary kind of folds back. Tertiary is a corkscrew. And quaternary is actually globular. It's like a whole bunch of primary, secondary, and tertiary all thrown together. It's a massive molecule. I will go over this again on the board, but you should be aware of it. Protein formation, whether it's a dipeptide or a polypeptide, it's going to go through the dehydration synthesis. We did this in lab, so you can refer back to that macromolecule or molecular model kit lab. And we also have hydrolysis, which is the reverse. Again, reference your textbook, reference your lab. The last item before we talk about the food sources is your protein indicator test. This we're going to do on Thursday and Friday. Biurettes, you're going to take, and actually we use biurettes, not the cupric sulfate and the sodium hydroxide. It will turn from a light blue to a purple. or lavender if it's protein is present. The other one is a nitric acid, clear and turns purple if protein is present. And obviously you can see those pictures. The last thing in proteins we're going to look at is the food sources. Now food sources, most people say, oh, you have to eat the meats. Well, you can be a vegetarian and still get plenty of protein. Your nuts, your beans have plenty as far as sources of protein. But again, you have a variety. Eggs, protein, albumin is high in protein, and all of your different meat sources. The last compound of life we need to talk about is nucleic acid. These are long chains of nucleotides, and the nucleotides are comprised of 3 components: a 5-carbon sugar, sometimes referred to as a pentose, the nitrogenous base, and the phosphate group. We have had these on the board numerous times. Remember the functions on these: primarily for the storing of our genetic material. Also, it transfers the genetic code into our protein synthesis through RNA. And finally, it is a carrier of energy with ADP and ATP. Those are our primary energy carrying components. When we look at that nucleotide, the monomer for nucleic acid, again, pentose, that 5-sided structure, the nitrogenous base, which for DNA there are 4 of them: adenine, guanine, cytosine, thymine, and the thymine, when it changes to RNA, is replaced by uracil, and then the phosphate group. The phosphate and the pentose, or sugar, in DNA create the sides of that ladder. The last one there that you see in the bottom left-hand quarter is actually ATP, and you see the phosphates that are attached. Now the phosphates, when they are removed, that's where you get the energy released. Last question usually is, where can we find these nucleic acids? Well, they're in every cell, but the best place that we can extract them without problems are your fruits and vegetables with seeds that you consume, your nuts and whole grains. You want to make sure that you have as much as unprocessed as possible. Now the unprocessed, when you're looking at that, it just makes it easier for us to get to those nucleic acids. If you have any questions, please make sure you note them, because we will fill them in, any questions, in class.